Hello friends, welcome to photosynthesis once again. Before to go to this point, just see some references. This is a light spectrum. Here you observe short wavelengths of light and a longer wavelength of light. This one is the visible spectrum. So visible spectrum ranges from about 390 to approximately 720 nm. In shorter wavelength, there are cosmic rays, X-rays, ultraviolet rays, while in longer wavelengths, there are infrared lights, microwaves, radar, radio, broadcast band. Again, if you see the visible spectrum, this one again the figure of visible spectrum. This is a visible spectrum 390, 380 to 750 is considered here. Violet, indigo, blue, green, yellow, orange, red, and after red there is a zone of far red light which ranges in between 700 to 750 nm. Today's point is about red dwarf, Emerson effect, and two pigment system. If one wants to see the mechanism of photosynthesis, then he must know what is red dwarf, what is Emerson effect, and what is two pigment system. But before going to red dwarf, Emerson effect, and pigment system, just say about the quantum yield. Quantum yield is nothing but the number of oxygen molecules released per light quanta absorbed during the process of photosynthesis. As you know, the pigment molecules are present. These pigment molecules act as an antennae, trap the solar energy in the form of quanta. After receiving the quanta or photon, the pigment molecules send that energy by means of resonance to the reaction center, and this reaction center or chlorophyll A, due to excitation, emits an electron. So this emission of one electron requires two quanta energy. Okay, so photosynthesis is considered as a two quanta process. That means it takes two light quanta energy to drive each electron. Okay, since 4 electrons are required for the reduction of one molecule of carbon dioxide, 8 quanta will be required to reduce it or to evolve one molecule of water. Okay, so what is quantum yield? Number of O2 molecules released per light quanta absorbed. Here we have to say about the number of oxygen molecules released per light quanta absorbed. When the one molecule of oxygen is released, when we consider this one reaction, when 4 water molecule undergoes the process of photolysis, it produces 4 protons, 4 electrons, 2 water molecule, and 1 molecule of oxygen. And for reduction of a carbon dioxide to carbohydrate, 4 hydrogens are required. That means when CO2 plus 4H, it gives rise to CH2O, that means C6H12O6 plus H2O. So here, since 4 electrons are required for the reduction of one molecule of carbon dioxide, 8 quanta will be required to reduce it or to evolve one molecule of water molecule. How this splitting takes place? If you consider 4 water molecule, then the splitting will give rise to 4 protons and 4 hydroxyl ions. These are the 4 protons, these are the 4 electrons, and these are the 4 OH. When 4 OH we consider, it gives rise to 2 water molecule and 1 O2. we are going to see about the red drop. So Emerson and Lewis worked on a quantum yield of photosynthesis in a monochromatic light. What is monochromatic light? Monochromatic light is nothing but the single colored light. This pair of scientists were working on the quantum yield, and they used different colored light to measure the quantum yield. Okay, they observed that this quantum yield, suppose this is a see this graph, this is a quantum see this graph, this is a graph of a quantum yield. These are the wavelengths, okay, and this one is a quantum yield. Up to 680, this quantum yield is a continuous one, but after 680 nm, this quantum yield decline suddenly in the red zone. In 680 nm, there is a red color is present, and as this quantum yield decline occurs in the red zone, therefore it is called as a red drop. Okay. So Emerson and Lewis were working on the quantum yield of photosynthesis monochromatic light of different wavelength. They observed that quantum yield declined sharply at a wavelength greater than 680 nm in the red zone, and this decline is called as a red drop. Emerson and his co-workers later on found that the reduction in the rate of photosynthesis due to red drop can be recovered by supplying additional short wavelength light along with the far red light. So far red light, that means the wavelength greater than 700 to 750, and short wavelength, that means these parts, these wavelengths. So Emerson and his co-workers found reduction in the rate of photosynthesis due to red drop can be recovered by supplying one more additional wavelength of light of a short wavelength. So this is a far red light and this one is a short wavelength. When we supply these two light at a time, then the problem of this red drop gets recovered. So this one is called as a Emerson's effect. This is a red drop. The decline in a quantum yield in 680 nm zone, that means red zone, is called as a red drop. And when additional short wavelength light is applied along with far red light, then this decline recover, again photosynthetic yield continues. This one is called as a Emerson's effect. So Emerson and his co-worker found that in the rate of the reduction in the rate of photosynthesis due to red drop can be recovered by supplying additional short wavelength light with the far red light. The monochromatic far red light, when supplied with the shorter wavelength of light, enhance the photosynthetic yield and recover the red drops. This enhancement of photosynthetic yield due to combination of light of shorter wavelength plus far red light is known as a Emerson effect. So Emerson's experiment confirm that at least two light sources or two systems might be present there, and these systems are called as a two pigment systems or photosystems or pigment systems. Okay. With the discovery of red drop and Emerson effect, it was concluded that at least two pigment systems are involved in the photosynthesis, and these two pigment systems are referred as a PS1 and PS2, pigment system 1 and pigment system 2, a group of photosynthetic pigments which absorb light energy and convert it into a chemical energy through a reaction center is called as a photosystem or pigment system. Group of photosynthetic pigment where present, they are present in the quanta zone, which absorbs light. Of course, quanta zone, the pigment present in the quanta zone absorbs the light and convert it into the chemical energy in the form of ATP and NADPH2 through the reaction center is called as a photosystem or a pigment system. The presence of two such a system has been supported by the studies based on chloroplast fractionation process, which showed two types of the particles within the chloroplast membrane. In a pigment system, a special chlorophyll molecule present in the center as a reaction center is called as a core complex. Okay. In a pigment system, a special chlorophyll molecule, it may be chlorophyll A680, that is, which is generally considered as P680, or chlorophyll A700, that means P700, is present in the center of reaction, is present in the center as a reaction center, and these are called as a core complex, generally denoted by letter CC. And is surrounded, that means the reaction center is surrounded by a number of accessory pigments. These accessory pigment and electron carriers called as a light harvesting complex. So there are two pigment systems are present in the quanta zone, pigment system 1 and pigment system 2. What is the difference between pigment system 1 and pigment system 2? Pigment system 1 has a reaction center chlorophyll A670, 680, 710 or chlorophyll A700, but most of the time P700 acts as a reaction center for the pigment system 1, while pigment system 2 has a reaction center P680, chlorophyll A680 is being the reaction center. What is present other than the reaction center? Chlorophyll B and beta carotenes are present in both the pigment systems. PS1 complex, pigment system 1 complex, consists of 200 chlorophyll, 50 carotenoids, 1 cytochrome B, 1 plastocyanin, 2 cytochrome B, 563 ferredoxin reducing substance, and it is rich in chlorophyll A, iron and copper, and they are present mainly in the stroma lamellae, that means intergrana lamellae, the lamellae which connects the two granum region. Pigment system 2 consists a colorless chlorophyll A, which is called as pheophytin. So pheophytin here acts as a primary electron acceptor. Pigment system 2 also consists of approximately 200 chlorophyll, 50 carotenoids like the pigment system 1. Primary electron acceptor here considered as Q, but it is now pheophytin. Then plastoquinone is present, 4 plastoquinone equivalents are present, 4 Mn molecules bounded to one or more proteins are present, 2 cytochrome B559 present, 1 cytochrome B, then manganese, calcium, chloride ions are also present. Pigment system 1 is present in the intergrana lamellae, while this pigment system 2 is present in the granum region. It involves only in a non-cyclic photophosphorylation, while pigment system 1 is involved in a cyclic and non-cyclic photophosphorylation. It is responsible for the photolysis of water and release of oxygen. pigment system second is responsible for the photolysis of water and release of oxygen, while such a type of photolysis of water or molecular oxygen is not liberated in the pigment system 1. It accepts the electrons released during the photolysis of water. Here, as photolysis of water is absent, therefore they will not accept the electron released from the water molecule. It helps pigment system second to produce assimilatory power. Okay, assimilatory power, that means the power generated during the process of photosynthesis in the form of either NADP, H2 or ATP, and pigment system 1 the particles are smaller and lighter one, while in pigment system second large and heavier particles are present. So this is about the pigment system, then red drop and Emerson effect. We will see the next part of this of the video in the